primary cilium is an antenna-like structure that is found on the surface of most cells in the human body. We know that these primary cilia are essential for mediating communication between the different cells in the body. So when the cilia are defective, the communication between the cells is perturbed or disturbed, and this causes a lot of different diseases that can affect nearly all the different organs in the body, both during development in the embryo as well as in the adult. In the sonic hedgehog signaling pathway, which is one of the most important signaling pathways in our body, both during development and in the adult, there are two membrane proteins that are key to activation of this pathway. And their ciliary localization is very dynamic. In the absence of sonic hedgehog ligand, so when the pathway is turned off, there is a receptor protein in the ciliary membrane called patched. When patched is in the cilium, it keeps another receptor called smoothant out of the cilium, and this is why the pathway is off. When sonic hedgehog ligand is present, it binds to patched in the ciliary membrane. This causes patched to exit the cilium, which in turn triggers ciliary entry of smoothant. The ciliary entry of smoothant then initiates a series of downstream events that culminates in activation of the signaling pathway. And this is really exciting because we know caveolin binds cholesterol, and in other studies performed by other groups, it has been well established that cholesterol is really critical for activation of the sonic hedgehog signaling pathway and for regulating this very first step in the pathway. So we believe that caveolin is really a key player in activation of this pathway. So even though we know that caveolin is really important for regulating activation of the sonic hedgehog signaling pathway, we still do not completely understand how caveolin itself is regulated at the cilium. We found in our study two other proteins that are important for regulating caveolin in the cilium. These proteins are called nephrocysteine 4 and KIF13B. Now, nephrocysteine 4 is known to cause kidney disease when mutated, and in fact, many other proteins similar to nephrocysteine 4 that are present at the base of the cilium cause various diseases when mutated. So in the future, it's really important to understand how caveolin localization and function in the cilium is influenced by all these other proteins that are present at the base of the cilium and that we know cause disease when defective. Currently, there's no drugs to treat these different cilia-related diseases, but there are some interesting studies that have been done in, for example, cultured cells, where at least two of the diseases caused by defects in cilia and hedgehog signaling have shown promising results with drugs that affect the sonic hedgehog signaling pathway.